All right. Hello, Biology 454. Yeah. um, I'm going to record the lectures so that you can go back to them. In general, what I'm going to be doing is just like talking about stuff in the book, and I'll make a lot of analogies. And the analogies are never, there's sometimes a useful way to think of something to help get you to the actual thing, but they're usually not, the best actual answer. They're not what you want to be putting down in the tests or memorizing. It's just a thing that if it works for you, then good, that'll help. So, like, for example, if I say that, oh, T cells they're kind of like enforcers or something like that, and NK cells are like whatever. That's fine if it helps to paint a picture in your head. What T cells actually are lymphocytes, which means cells that are flowing in the blood that are not erythrocytes, like the red blood cells. Okay, great. And they come from a particular stem cell population. They have various functions, various proteins on the surface. I mean, that's the reality. So this is to try and help get you to that reality. So I'm not going to go over all the definitions because you can read faster than I can talk and better than I can talk. I'll go through what is mostly going to be the pictures or anything that I think is interesting. And the idea of this is just to give you another way to study these things. And at some point, each of these little bits is going to click, and then you're going to say, that's fine, I've got it, I understand, and we can move forward. Because everything that we're doing in this half of the class is very much going to build on everything that comes before. So we got to make sure this foundation is rock solid and steady. So that's what I'm trying to do. That's why we're here. And so here you go. This is for you. Enjoy, I guess. I don't know. Fine. So we're going to be talking about T cell receptors and B cell receptors and how did they get the way they are. All right, here we go. Fine. We don't need that, and we don't need that, and we don't need that. I mean, those are all good things, but they're just not stuff that I'm going to read to you because you're good at reading. Oh my gosh, yeah. You've been good at that for years. So, behold the antibody. Now, I don't think it says which type of antibody this is, but this looks like it could be either an IgG, so immunoglobulin type G, it could be an IgD, or it could be an IgE. Big difference between those is the number of disulfide bonds here. And honestly, I don't remember which one has the two. I suspect this is an IgG, but yeah, whatever. You can look up the answer and then you'll know. This thing rotating above me is just for visual appeal. It is an IgM molecule. So you can see IgM actually is like 5 antibodies with their Fc regions sort of stapled together. And it is interacting with another protein that looks like an immunoglobulin-like protein. So that may be another antibody. I don't know. I have to go back and read the paper to see what that is. But yeah, there you go. So in our antibody, we're going to have 2 chains generally, which is going to be heavy chain and light chain. So 2 types of chains. And by chain, I mean this is actually a protein that comes from a messenger RNA and is translated in one piece and then goes on to fold up and have a function. So yeah, it's not literally a chain. I don't know. That's just what everybody says. But they call them heavy chain and light chain. Awesome. In the heavy chain and in the light chain, there is a region that is constant, which basically doesn't change, or the changes are very few and very minor. And it's basically everything except the top little bits here. So the variable region is this little VL thing. The variable region of the light, and the VH is the variable region of the heavy chain. So our heavy chain is just like twice as big as the light chain. You can see this purple bit is one big long protein that folds up into 4 immunoglobulin-like domains. And the light chain is just 2 immunoglobulin-like domains, and it's going to sit right next to this and interact with the heavy chain. And the tips of those, these sort of variable domains, at the top of the antibody, that's going to be where we get our antibody specificity. Like, that's the thing that's going to make one antibody different from another antibody. And like an antibody against the common cold is going to have a different set of amino acids on it than an antibody against, I don't know, E. coli or something like that. Yeah. All right. So the only other thing here is have a look at that immunoglobulin-like domain. It basically is consisting of beta strands that are sort of stapled together in beta sheets. So think of my fingers as strands, and now they're a sheet. Great. I don't believe you can ever have just one beta strand on its own in this world. I believe you need two of them together, either like pointed the same way or a different way. So beta strands only exist in sheets of two or more strands, but they can be very large as well. And they can sort of twist a little bit in the sheet, and that's fine. It's still a sheet. It doesn't have to be planar, like flat. Okay. That's enough structural biology, right? Yeah. Okay, heavy chain, light chain. Cool. The light chains are held onto the heavy chains by disulfide bonds. A disulfide bond is two cysteine amino acids, cysteine, and they have a little sulfur, SH is their side chain. Sulfur and a hydrogen. And they're going to kick out those hydrogens, and the two sulfurs are going to bond to each other. And that's a disulfide bond. They can be broken by chemical agents, but disulfide bonds are generally made in regions of the body where the environment will preserve them. So basically, a disulfide bond is like a permanent, well, it is a permanent covalent linkage. You can reverse it in a chemistry lab or something, but in biological reality, it is rarely going to be reversed, except when you're like recycling a protein. There would be ways to do it then. All right. Boom, boom, boom. So yeah, there's our little immunoglobulin domain. And so you can see at the top of this thing, here we go, we've got these little loops. And the little loops are going to be, and by loop I just mean the sort of curly bit in between two strands. So here's our strand, there's our little arrow of the beta strand going up, and then it's coming back down, this big long one. And so this thing where it sort of turns, you can call it a turn or a loop if it's like anything bigger than the minimal amount needed to sort of flip and go back the other way. These are the parts that are going to be hypervariable regions. So they're going to vary a lot. Every single antibody is going to have something different going on up there. But antibodies are all proteins. And proteins are only made of 20 common amino acids. There are situations where you can get other rare ones in there, but mostly those don't apply. So just put those out of mind for the moment. And so antibodies can bind almost anything that amino acids are able to bind. And it's good because you have amino acids that have these different side chains on them. So some will be acidic, some will be basic at particular pHs, some will be hydrophobic, some will be kind of hydrophilic. And so there are a lot of ways that they can interact. They can hydrogen bond, they can do all kinds of bonding, ionic interactions. The only thing you won't see an antibody doing in general is covalently bonding to something. So they're not going to, because that would be like a reaction. And if you think of it, that'd be pretty weird, because stuff that reacts would be things like free radicals, for example. That's just a molecule that once it's broken, you have an oxygen with an extra electron, and it just goes off and reacts with something, like the next thing it runs into. In the chemistry lab, you control these reactions by only having a couple of things there in the flask that can even possibly react. But a cell is big and complicated, and if you have literally reactive things floating around in there, you're just going to it's going to be a mess. Yeah, the cell is not going to make it. And so that's why cells generally use enzymes. That's kind of like a gun with a safety. An enzyme has to have various things plugged into it and various energy sources docked in and just the right molecule that fits in. It's just got a lot of, requirements. It's got a long list of things that have to happen before it will do a piece of chemistry for you. And then they're pretty specific on the chemistry that they do. So, yeah. Yeah, I think that makes sense. Hope it does anyway. I don't know. You'll have to tell me. Yeah. All right. And so there are various parts of the antibody. The top bit is the antigen binding fragments, the FAB, and the bottom bit is the FC, which I believe is the constant fragment. And that's fine. So when you see these words up here, like amino terminus and carboxy terminus, do you know what those mean? I mean, yeah, it depends how much protein biology you've had up to this point, I think. The amino terminus is the front end of a protein. The carboxy terminus is the tail end of a protein. So carboxy is the caboose, if it's a train, yeah. And amino is like N, which is like the engine, yeah. It's stupid. Don't ever put that on a test, but if it helps you remember it, whatever. You don't have to tell anybody. This is the way you're remembering things. Yeah. All right. And FABs and FCs, these don't exist separately in the real world. These are things that immunologists made. You can take an antibody and you can cut it, and it just happens to, that a common protein cutting enzyme called pepsin or papain will, pepsin will shred it, but papain will just cut it, kind of nick it. It happens to cut between these two parts, and so it's like a historical thing. The FC is what we call the bottom region. So the FC is what the cell is going to hold. If you imagine this as a beautiful fork, these are the little pointy ends that are going to be stuck in the food, or in this case, they're going to stick to the antigen. The bottom is the bit that the user is going to pick up, which is going to be some type of cell generally. But you would also have things like complement that could bind to an FC that is bound to some sort of target. So there's different ways to recognize. So the FAB recognizes foreign things, and the FC is recognized by you. That's like your special little badge that this is one of my antibodies. And so when people make therapeutic antibodies, which, by the way, most of those, if you see a new drug and it ends in MAB, that's going to be a monoclonal antibody. Yeah. So something IMAB or UMAB or whatever. Yeah, that's just how they name all of them. So yeah, look for it, and all of a sudden you'll start seeing them all over the place. A lot of people like these. All right, and antibodies don't do anything. Antibodies just stick to things and allow other cells and other proteins, enzymes, whatever, to do their thing. So an antibody is an adapter. It connects a foreign pathogen, even an unknown foreign pathogen, to the rest of your immune system so that you can deal with this thing, which, yeah, is a pretty good idea now that you mention it. Yeah. And is also the way that everything works. So, I guess it doesn't matter whether you like it or not. It's just reality. Yeah. Fine. So cool thing about antibodies is that even the simplest antibodies will have two little FABs, so kind of like that. And so they'll have two little antigen binding sites, which means that if you were to circle like this antibody in the bottom corner of the triangle, you can see it's holding on to two separate antigens at the same time. And so like here's a little triangle here. This is actually an electron microscope picture of antibodies holding on to antigens. So you'll see some triangles in there. You'll also see some squares in there, which are going to be 4 antigens held by 4 antibodies. And you'll see what looks like a pentagon down here, which is probably 5 antigens and 5 antibodies. Look at that little squished hexagon. That could be 6 and 6. And yeah. So antibodies can both neutralize multiple antigens at the same time. They can hold on to multiple antigens. And so if it's like a little thing that could float around freely, like a virus, then one antibody can rope 2 of those together. It's just like, yeah, I don't know, tying 2 dogs together. They'll probably both stay a little closer unless they can agree on what they want to do. And also this whole thing is called an immune complex. So it's a whole bunch of antibodies stuck together with antigens. So this is another thing that can form. And certain cells in the immune system can recognize immune complexes with a whole bunch of antibodies together. And they'll have special responses for that. They'll either sort of bring them in and digest the antigen to see what the heck is this, and can we show it to the T cells and, get a good response going, or yeah, just react against it. All right, so first section done. And this is in 3 sections, and I may be able to knock them out all in one video. I don't know. Actually, no, let's be sensible about this. I will break this into 3 little mini videos, and so that way you don't have to put bookmarks in, because I sure don't know how to put bookmarks into YouTube videos. All right, good enough. Yeah, see you.